Hello, good evening, and welcome. I'm going to talk about biology, which I think probably nobody else is going to. Well, I hope not. I want to try and be, unique. Okay, so we can get started. As you may see, this is Craig Venter. He is basically the patron saint of genetic engineering. He is one of the men who are behind the whole Human Genome Project. Bonus points for anybody who can recognize the plasmid that makes up his halo, which I was very proud of. And all of his efforts led to being able to identify all of the information that's encoded in what you're able to see here, which is basically, it's the typical karyotype for a human genome. Okay, so a genome is a collection of all of the genetic information that's actually inside an organism. And what we're able to see here is that that genome encodes for tools, different types of molecular objects that do jobs. And this is the proteome. These are all proteins. They all basically do anything that's happening inside the cell. Here we have a stock photo of a woman doing genetic engineering. And what she's doing is she is cutting apart the chains of the DNA, and here she is inserting something else into. I think it's the same woman. I'm not sure. Here she is inserting information into a genetic code. Basically, genetic engineering, overall allows us to manipulate the information that's actually available for organisms. Okay, so synthetic biology likes to paint futures where we have things like these, which are zebra roos or whatever. But really, at the end of the day, the actual output is actually more like this, where we have mice that have green fluorescent protein encoded into the keratin that's in their bodies. Okay, so it's part of the actual connective tissue itself, and that can help to highlight structures within the body that are important for us to actually look at. Now, synthetic biology is really interesting because it uses a lot of computer metaphors in order to understand how it actually operates. So we have tissues and cultures and so on, and they're the networks, all the way down to biochemical reactions, which are the gates and the actual information processing that goes on. And these computer metaphors are really, really handy to actually teach people who don't have any kind of background in biology about how these biological systems actually operate. Now, what I like to highlight is, and this is something that's important for actually, when you're actually thinking about it and how you're approaching it from a design perspective, is that every organism is actually an emergent system, okay, from the interplay between the environment and the actual genetic information. So, this landscape here and a specific genetic information gives rise to this, basically the Fabio of horses. When we have these organisms, we need to not just control the information that they have, but control how that information is expressed. Okay? So, in one way, with synthetic biology, we have the top-down approach. Okay? So we have bacteria that are already evolved to do a specific job, okay, and they're kind of munching along and dividing and so on and so forth. And what we do is we take DNA from something like a human cell, and we put it into that bacteria, and that bacteria then does a new job. Here you're able to see humulin, which was one of the first synthetic biology-derived products. Basically, it's identical to human insulin, except that it's extracted from these little bacterial refineries. The other side of the story that we're able to go from is the bottom-up approach, where we approach it from protocells. Okay, we start out with something that looks like something even before human cells or mammalian cells differentiated from bacterial cells, and that's how Craig Venter, who I mentioned at the beginning, created Cynthia, which was the first fully artificial cell. Okay, so I like to be on top for the top-down approach. Okay. I like to use creatures and systems that are already there to do new jobs. Okay, get them running, because they already know what they're doing. We just want to push them in a different direction. And I also like to get in trouble. Okay, so I don't believe that genes and genetic information should be able to be patented. And this has led me to the discovery of a project that I've been working on for the last 3 months. It's a tribute to two artists, Shishio and Georg, who will hopefully see this video at some point. This is basically taking a genetically modified carnation, which is sold to us with the idea that it will never seed. We'll never get access to the seed. It's sold to us as cut flowers. It dies after 3 weeks. What we can do is we can take cuttings, okay, and using a very specific media, we can grow these cuttings from the axial buds and the stems into new differentiated plants. So we can actually take that genetic information from where it's actually stored in the stems, and we can grow a completely new crop. This allows us to steal this information. It's basically genetic hacking. And with it, we can change everything. Thank you.